गुड मॉर्निंग स्टुडंट. नवयोग शिक्षण प्रसारक मंडळ चापोली संजीवनी महाविद्यालय चापोली वेलकम यू इन e-learning platform prepared by myself Dr. Shubhangi Prabhakrao Chavan, Assistant Professor and Head Department of Botany. Now, Swami Ramanand Tirth Marathwada University Nanded Semester Pattern Curriculum under Choice Based Credit System CBCS for undergraduate program, Faculty of Science and Technology, subject Botany, year BSc second year, paper Plant Embryology. Now, student, in last lecture we see basic of embryology. Today we will discuss microsporangium structure of anther and TS of anther. Now, start microsporangium structure of tetrasporangiate anther type of tapetum sporangious tissue. Now, first of all, introduction. Androecium is the male reproductive and essential oral of flower. The male reproductive part of flower is called as stamen. It consists of parts like filament, connective and anther. The anther consists of microspores or pollen grain in a sac-like structures or chambers called microsporangium. In angiosperms, the anther are monoecious, that is the one lobe, example family Malvaceae. Now, plant hibiscus or Dathonius, that is two lobes, example most of angiosperms family Rutaceae, Solanaceae, Acanthaceae, so on. The anther with one lobes consists of two microsporangium or two chambers, thus is the bisporangiate. The anther with two lobes consists of four microsporangium or chambers, two in each lobes, thus it is referred as tetrasporangiate. Now, in figure first, we observe monoecious anther. Next, we can figure second show different parts of the stamens. We can see in figure A, first of all, filament, anther, lobes and structure. Next, figure B show the dorsal view. In this, connective can be noticed clearly. Next, figure number C, TS show pollen chambers and pollen grains. Now, next, each lobes of the anther bears two chambers which produce microspores or pollen grains. Hence, it is known as microsporangium. Next point, structure of anther. Anther has two anther lobes. Each anther lobes show two chambers. Hence, anther is known as tetrasporanginate anther. Each microsporangium at an early stage process sporanginous tissue 2n, which gives rise to microspores mother cells. Sporangious cells divide mitotically several times before functioning as pollen mother cells to increase the number of functional pollen mother cells. Later, and another changes in size, hence pollen sac also increase in size. Functional pollen mother cells which are diploid to N produce a mass of haploid N pollen grain through meiosis. Now, next, non-functional pollen mother cells degenerate and provide nutrition to the developing pollen mother cells. The process of development of pollen grains from functional pollen mother cells is known as microsporangiasis. The anther is made up of single lobe or two lobes. When another consists of single lobes, it is called monotheous. In majority of angiosperms, their anthers consist of two lobes called diothes. Now, next, development of microsporangium. First of all, initially the anther consists of only a mass of homogeneous tissue which are meristematic in nature. The sum of the meristematic tissue below a protoderm becomes special and prominent and acts as archaeosporium or archaeosporal initial cell. Archaeosporal initial cell divides periclinial to form two layers, that is primary peritial layer and primary sporangious layer. Now, next, the primary peritial layers divides periclinial and anticlinial and form the wall layer anthers. The primary sporangious cells either directly function as spores mother cells or divide mitotically into survival cells which function as spores mother cells. The spores mother cell undergoes mitotic division and gives rise to four microscopic arrangement tetrahedrally. Now, next, figure discuss. Now, describe stage of anther development. First of all, TS of young anther. We can see epidermis and meristematic cell. Next figure, differentiated archaeosporial cells. Now, we can see archaeosporal cells and procambial strength. Now, next, differentiated primary peritial cell and primary isosporangious cells. Now, we can see epidermis, primary peritial cell, primary sporangial cells. Now, last, figure number D. These are epidermis, anther wall which can endothecium, middle layer, tapetum, spore mother cells. This is the development of anther. Now, next, flow chart of development of pollen grains from anther. First of all, anther, epidermis, archaeosporangium. Now, archaeosporium, there are two subpoints, peritial tissue, sporangial tissue. Now, peritial tissue, endothecium, middle layer, tapetum, and sporangial tissue, therefore, pollen, MC means mother cells. Pollen create pollen grains Now, next, structure of tetrasporanginate anther. Now, this is the discussion of, let us discuss in detail structure of transport tetrasporangiate anther. First of all, this is the TS of tetrasporate anther. In this figure, we can clearly observe all parts of anther start from upper layer epidermis, then endothecium, middle layer tapetum, sporangious tissue, spores, statutum, connective tissue. Now, next, structure tetrasporanginate anther. Transverse section of anther show follow well layers. First of all, epidermis, next endothecium, next middle layer tapetum, sporangious tissue. First of all, first point, epidermis. Now show epidermis is the outermost layer of the anther. It consists of a single layer of cell, and cells are compactly arranged. It exists from the very early stage, and it is not derived from the primary parietal layer. The cells of the epidermis undergo anticlinear divisions, and so the epidermis increase in surface area. Now, next, in mature anther, the cells of epidermis are greatly stretched and flattened. Epidermis as protective in function. Next point, endothecium. Now show the figure microsporangium showing endothecium. We can see inner tragedial wall, outer tragedial wall. Now, endothecium is the second layer of anther wall and is also known as fibrous layer due to the presence of fibrous bond. It is present just below the epidermis. It is from primary parietal cells. Now, it consists of single layer of radially elongated cell. The endothelial cells show the characteristics fibrous bonds and linking with areas from the inner tragedial walls. Now, next, the cell of the endothecium along the line of detentions of the anther divide of the fibrous bond. These cells are called stemium, where the endothecium is thin-walled. The stemium helps in detention of the anther. The endothelial layer become hygroscopic due to the development of fibrous bonds, and they play an important role in antheresis. Endothecium is absent in anther where distant decisions is through apical pore. Example, cassia, selenium. Fibrous bonds are absent in apomia, anona, mesua and apomia. Now next, middle layer. Middle layer, it is the third layer of anther wall lining below endothecium. It consists of 2 to 3 layers of thin walled parenchyma cells. It is known as peritial layer. Now next, the cell of the middle layer store starch grains. The middle layer cells get crushed at the time of meiosis in pollen mother cells. Next point, tapetum. Tapetum is the innermost layer of the anther wall. It develops from the primary peritial layers. It is formed beneath the middle layers. It is formed of a single homogeneous layer surrounding the sporangious tissue. The cells of tapetum are large, radially elongated and continuous, dense cytoplasm and protomium nucleus. Tapetum is nutritive layers for the developing microspores. Now, structure of microsporangium showing tapetum. We can see epidermis, endothecium, middle layer, microscope mother cells and tapetum. Next point, sporangious tissue. The sporangious tissue are formed from the archisporial initial cells or archisporium directly. The formed of cell directly act as pollen mother cells or spore mother cells, PMC, SMC. The pollen mother cells are diploid 2n in nature and they undergo meiosis to produce pollen grains, N, PG microspores. The pollen grain in further development produce male gamete. Now, next, tapetum. Vascularly, the tapetum is peritial in origin, but in elytra thymisin, the tapetum is dual in origin. Protein of the tapetum, which is derived from the cell of the connective tissue, are called C tapetum, and the remaining protein of tapetum is derived from peritial cell and called P tapetum. There are two types of tapetum. First of all, C tapetum and P tapetum. These cells are the C tapetum are larger and the cell of P tapetum are smaller. The type of tapetum with two different types of cells is called dimorphic tapetum. Now, next, types of tapetum. Based on the behavior of tapetum, it is two types. First of all, glandular or secretory tapetum, and next amphibia or plasmidial tapetum. Glandular or secretory tapetum. It is also known as secretory or peritial tapetum. In glandular tapetum, the tapetal cells remain intact in their original position throughout the development of microspores. The tapetal cells don't deteriorate, the cell become rich in protoplasmic con- contents and secretes a plasmidium that surrounded the pollen grain. They secrete their substance from their inner face. They process almost all cells originals like mitochondria, plastid, decto, dectosomes, etc. Secretory or glandular tapetum is common than the amphibia tapetum. It is found in the anther of helibeals, fadils, metibeals, and jumper. Now, just before the pollen mother cells undergo meiosis, the wall of the tapetal cells become thick and there it considerable increase in the number of ribosome and bodies which helps in development of pollens. In helobials, in helobials, uh, folials are Echen and Godwin have recorded the formation of proteus bodies in the tapetal cells. These bodies contribute a material known as sporopollen, which they involved in thickening or exine of pollen grains. Example: higher monocot and dicot. Amphibian plasmoid tapetum. It is also known as invasial or pre-plasmodial tapetum. In amphibial tapetum, in the inner and radial walls of the tapetal cell break down due to the hydrolytic enzymes and the protoelastic becomes a plasmodial mass called tapetal periplasmodium in which the pollen mother cells are suspended. Plasmodium remain associated with the pollen grain till then the maturity. This type of trapedium considered the primitive types. Amphibious types of trapedium is found in anthers of uh, alisma, tripha, tetroscancia, butamus, Sageria, Potomagoton, etc. In both amphibia and glandular tapetal cells, total DNA content increases during meiosis. As a result, the tapetal cells may be uniculated, biculated, or colliculated. Function of tapetum. Tapetum is the innermost prominent layer of anther wall. It attains the maximum development of the tapetal stage of microsporangiasis. The important function of tapetum are follows. Tapetum is native tissue in the developing microsporangium. The tapetum from the channels for the transport of materials to reach to pollen mother cells. Now, next, the tapetum serve an important role in the pollen wall formation. The unrich bodies present in the tapetal cell considerable of materials called sporopollenin, which is involved in the formation of exine of the pollen grains. This has been demonstrated in helobials, fabelials, by Eachin and Godwin 1968. The plastid of the tapetal cells synthesize material like pollen kit of tryphin uh, discount in 1978. These material protect the pollen grain from damaging effect of ultraviolet rays, and it's strictly nature helpful in insect dispersal of pollen. The enzyme callus, which is derived from dictosomes of the tapetal cells, degrades the callus around the pollen trait, and hence the pollen grains in the traits are separated. This has been demonstrated in the tetrascansia by the Mayfawn Lane Mayfawn and Lane 1969. Tripetium is essential for the formation of herbal pollen grains. Tetital degeneration during the mitotic stage, causal male sterility in planua. These proteins present in the exene